Hello, this is the second video of the respiration topic, which is topic 3.3 of biology, and we're going to do the next part after we've done glycolysis last week. This is now the link reaction and the Kreb cycle. And I just wanted to say about what we did last week as well. Glycolysis won't be making lots of sense to you at the moment in terms of understanding why things are happening or what it's happening for, or being familiar with the molecules, and that's okay. With this respiration topic, there's the whole process of glycolysis, the link reaction, the Kreb cycle, and then the electron transport chain. And it's only really when you've learned all four of those parts of it that it can all come together and you can understand what the different roles are of the different molecules, how it all comes together, and what these different processes are. In the textbooks and in the specification, the first topic of this unit, 3.1, is about ATP. And I think some of that ATP topic is worth looking at before this, but a lot of it is worth looking at afterwards, because again, it's once you know the process of how it takes place that you can then understand about the different types of phosphorylation and what that means, because otherwise, without knowing this process, it doesn't make much sense. So knowing glycolysis just as a simple metabolic pathway, knowing what the molecules are and which ones you need to know and which molecules are made, where it starts and where it finishes, then that's great for now. Do the same with the link reaction of the Kreb cycle. When we get to the electron transport chain, after that, that's when you need to start putting it all together and understanding what it all really means. So don't worry if for now that's just a bit of learning and becoming familiar. We'll do the same with this, and then we'll be able to put it all together later on. Okay, so the link reaction does exactly what it says. It links the glycolysis pathway to the Kreb cycle. And it's just a sort of little bit in between. Because glycolysis takes place in the cytoplasm of the cell, the first part of the link reaction is just acknowledging that it brings the reaction and the pathway into the mitochondria. So first of all, we're entering the mitochondrial matrix. And the molecule that we start off with is this 3-carbon molecule, which is what we had at the end of glycolysis. So at the end of glycolysis, one glucose molecule was broken down, it was split at one point, and then split into these two molecules, which became 2 pyruvate molecules. So here we have one of those pyruvate molecules. So it's worth noting now, and we'll come back to this as well, that this all happens twice for every one molecule of glucose that goes through respiration, because of that split during glycolysis. So in glycolysis, that one glucose molecule became, after the process, the 2 pyruvate, and now we have one of these here. So this pyruvate molecule enters the mitochondrial matrix, and just a few things happen to it. It loses carbon dioxide, which is called decarboxylation. So CO2 is lost first of all, decarboxylation, and then it is also oxidized. So the molecule here, at this point, once the carbon dioxide is gone, this molecule isn't pyruvate obviously anymore because it's lost carbon dioxide, and we don't need to know the name of this molecule. So you just call that an intermediate, and we'll refer to intermediates as we go through. They don't expect you, the WJC don't expect you to learn lots of names of molecules here really. It's mainly what's coming and going, what's coming off, and rather than the intermediates you need to know. So the pyruvate is first of all decarboxylated, loses carbon dioxide, and then it is oxidized by NAD, which you're not very familiar with at all, but you met it in glycolysis. So NAD, I said, is a coenzyme that can carry electrons around. So in this case, it's taking electrons from this intermediate, which means that this molecule is being oxidized and NAD is reduced. So NAD becomes reduced NAD. Then we have a molecule here that is actually acetate. So acetate here, the 2 carbon molecule, because carbon dioxide has been lost, and it's been oxidized, so it's acetate. And what's added here is something called coenzyme A. Again, you don't really need to know what that is, particularly with coenzyme A, sometimes called CoA. So if you see it a lot of the time in videos and textbooks, it'll be called CoA. So coenzyme A is added. So what we have now is the molecule at the end is acetyl coenzyme A, or acetyl coenzyme A. You see it. So acetyl CoA output. So there's acetyl coenzyme A. And that's now the molecule that is at the end of the link reaction. So the link reaction starts with pyruvate from glycolysis, ends with acetyl coenzyme A, and that is the molecule now that is going to be entering the next stage, which is the Kreb cycle. The Kreb cycle is sometimes called the citric acid cycle or citrate cycle, but often I think that's more American that they call it that. So we've got this 2 carbon molecule here. So this we now know is acetyl coenzyme A. And this metabolic pathway, some people actually imagine it happening in a little wheel shape. Obviously, it's not happening in that shape. The point is that the reactions are occurring all inside the matrix. All these reactions are happening all at once, but each of these molecules will react, one after the other, and then the molecule that is here will join with that one, which is why it becomes a cycle. So the one I'm going to skip to the end in a way. We have a molecule here which is called oxaloacetate, and that is a 4 carbon molecule. Pop that one in here, ready, because we know that it needs to join with acetyl coenzyme A. So I'll name that one. Okay, oxaloacetate, you don't actually have to learn the name for that one. You can call it an intermediate if you like, but that's the one that I always use because I think it's useful to be able to name it, because it's useful to say that those 2 are joining together. So oxaloacetate there, that will join with acetyl coenzyme A. So obviously here now we have a 6 carbon molecule because the 4 add 2. So 6 carbon molecule here. Okay. And this one, and this is the last of the intermediates that I'm going to make your name, but this one is citric acid or citrate, and that's why it's called the citric acid cycle as well. So that there, we'll call it, there it is, citric acid. Okay. So what we've got then, I'll just go back through it briefly. We had a glucose molecule in glycolysis. A series of reactions took place. One of the parts of that was it splitting. It produced ATP, it reduced NAD, all those sorts of things, and now we have 2 pyruvate molecules here, like a 3 carbon one. That moved from the cytoplasm into the mitochondrial matrix. It lost carbon dioxide, which is decarboxylation. It was oxidized by NAD, which means that it lost electrons to NAD, which made NAD into reduced NAD. It gained a coenzyme A, so now it became this 2 carbon molecule, acetyl coenzyme A. Now still within the mitochondrial matrix, so we are, there's your inner membrane. So we're in here now. Inside the mitochondrial matrix, acetyl coenzyme A now joins up with oxaloacetate, which is been produced at the end of this metabolic pathway in the Kreb cycle. They join together and form citric acid. So citric acid, the 6 carbon molecule. Now, this, I think, looks complex to learn, but if you just learn it in a few steps, then it's not so bad. Okay, so first of all, we'll start off with just dealing with the carbon part of it first of all. So it goes from 6, if you just remember this, it's not too much this bit, it goes from a 6 carbon molecule down to being a 5 carbon molecule. And this is one of those intermediates, we don't even care what it's called, we don't have to know. So there's a 5 carbon molecule there. Okay, then here there's a 4 carbon molecule. And then again. And then again at oxaloacetate. So from that point of view, all we have to know first of all is that it goes 4 plus 2, which is 6, then 5, and then it remains at 4 carbons all the way around there. So by knowing that, if you remember that, then you know where carbon dioxide comes off. Carbon dioxide is obviously going to come off anywhere where you lose one of those carbons. So here we'll have carbon dioxide there and there, CO2. Okay, so that's the first part of it. Then the other part of it is to do with these cofactors here and those molecules that carry the enzymes. And they're the ones that I said are really important, that are going to make our ATP basically. So they're the ones that we're building up to this electron transport chain. So that's the next part. And we've got carbon dioxide coming off. And then the way that I remember the next bit, which is stupid obviously, is NAD, NAD, a FAD, NAD. So if we go from here, we've got, there he is. So put there. So NAD, NAD. Put this one down here. FAD. Now FAD is FAD now, which is a different one. Similar but different. And for now, that's fine. You don't need to know any more than that. And then NAD. And if we just finish this off and just add in these little details, which you don't really need to remember, you'll know these. That is all you need to learn of the Kreb cycle, and you'll get more familiar with what it means a little bit later on, like I said. So we have NAD oxidizing this intermediate, which means you get reduced NAD. Now, to save space, remember I said that reduced NAD is sometimes called NADH. It's picked up electrons, but it's also picked up a hydrogen. So I hope this doesn't confuse you, but I'm just going to call it reduced NAD, NADH. You do need to get used to it because it will be on other stuff as well. So NAD is now reduced NAD, and it has oxidized that intermediate. It's lost another carbon dioxide, and then again it's been oxidized by NAD, and we have NADH. I can't even fit it in like that. There, NADH. Then we have, I said it was NAD, NAD, a FAD, NAD. So the A is ATP being formed. So we had ADP, and then we get ATP. So we do make 1 ATP on the Kreb cycle. Then the FAD becomes reduced FAD, just the same as the NAD did. So we get reduced FAD, and again, you don't need to understand much about that. We'll talk about that much more, and what it is and what it does in the electron transport chain. I'm building it up, but it's my favourite bit anyway. So reduced NAD, or NADH, like I've just done the others. Okay, so hopefully it didn't look like too much to learn. I know it looks like, it does look complicated at first, but it's not too bad. So you get the acetyl coenzyme A, the 2 carbon molecule, that joins with oxaloacetate, which is 4 carbon molecule. So obviously 4 add 2, you get 6 carbon molecule. That one loses a carbon as carbon dioxide, so it becomes a 5 carbon molecule. Then again, we don't know the name of that intermediate, you don't have to know, loses another carbon dioxide and becomes a 4-carbon molecule. So remember that it goes 6, 5, and then 4 the rest of the way round. Then NAD, a FAD-NAD, and that is just that NAD oxidizes it. Again, ATP is formed, FAD oxidizes it, and NAD oxidizes it again. And if you can learn it even like that and not thinking too deeply into, like I said, what everything is and what everything does, we will get to the electron transport chain. It will make much more sense. Okay, so when we move into the electron transport chain, my next video will be about that. These molecules will be really important. You'll have to remember what's being carried forward from here, which will be these electron carrying molecules here, the NAD and FAD. You don't call them NAD or FAD, by the way, that's me being weird. So the process here, like I said before, just remember as well that this is happening twice for every glucose molecule. So respiration is about the breakdown of glucose for the production of ATP, and because during glycolysis we got 2 pyruvates, this whole thing happens twice for every glucose molecule.